Hello everyone, and welcome you all in CoronaCures webinar series. I'm Yogita from CoronaCures and Skype. As we all are aware of the severity of the current pandemic, its common causes and consequences, but the question arises when COVID-19 infects differently to the people from different age groups. Like older people are more vulnerable while children are not. So therefore, to address these questions, we have invited Dr. Anil Singh. Welcome, Dr. Anil.
Thank you.
So Dr. Anil is an assistant research professor at Washington State University. And he works on understanding the mechanism of autoimmunity in rheumatic diseases and the therapeutic potential in synthetic or naturally derived small molecules, particularly in rheumatoid arthritis and gout. In today's session, he is going to talk about COVID-19 and the cytokine storm and the critical role of interleukin 6. So basically, his talk will include the origin of cytokine storm in COVID-19 patients, related pathways, and targeted interventions against it. So before we start the presentation, I request our audience to please post their questions in the chat box, and we will try to address them at the end of the session. Now I request Dr. Anil to please start the presentation. Dr. Anil.
Thank you, Yogita. Thank you for wonderful introduction. And as mentioned by Yogita, I'm currently work as a assistant professor in Washington State University at Spokane. Spokane is a very moderately big town, and it's one of the largest city situated between the Minneapolis and Seattle. Between the Spokane and Minneapolis, there are almost like a 2,000 mile. There's only few cities exist, like Billing in Montana and all. And it's a wonderful place. It's one of the top 100 tourist destination in the United States. And Washington State University is one of the vibrant place to work, and it's one of the well-funded in the state of Washington. And so with this, here are a few of my declaration. And my declaration, although I don't need to show up declaration, but for the scientific talk purpose, I must mention it. So I have no financial interest for this presentation with organizer, which is the CoronaCure.co. And the presentation is purely for educational purpose with the intent to help scientific community and awareness. And there are many people who still have the question for them. And all points in this presentation are my interpretation of published data with unbiased approach. However, viewers can have their own interpretation. So don't get biased with my own interpretation. And since I'm not a virologist, so I can't say that everything happening inside humans, I know it. So I have a different perspective, and the perspective is again is similar to diversity because I come from a different background, and since I have a PhD in biochemistry, so I understand many things of the life at the biochemical level, and currently I work on a rheumatoid arthritis. So working on a rheumatoid arthritis make me in a unique situation because rheumatoid arthritis is autoimmune disease, and it's primarily involved inflammatory signaling, very high inflammatory signaling in the synovial microenvironment. And this inflammatory signaling is pretty much like a cytokine storm, which happens in COVID patients. And for some reason, I found very blessed because I was working on IL-6 trans signaling. And IL-6 trans signaling is very one of the hot topic in rheumatoid arthritis. And I have some data which is going on with the IL-6 trans signaling related with how fibroblast functions and how differentiation like reprogramming happens in rheumatoid microenvironment. And I found opportunity here when Priyanka approached me, and I said, hey, I can talk about this because I have many content. So So with this, I'll present my case. And so with the COVID-19, which is reported in December 2019, confirmed by China on 7th of January 2020. And when it showed up, it's a chaos. It's a chaos because people thought, what if it's like a MERS and SARS? And eventually we found out it's actually worse than that. So what actually changed with this COVID-19? In 2020, everything was fine in January. We were spending our time with family in Las Vegas, and we were all roaming around, gathering here and there. But what changed actually? The Suddenly with COVID entry, there are many crises happen in the world, political throughout the world, and most of the time it's denial. And now we are in the threat of sinking. The world is sinking. Already the world was drowning with the debt, like $27 trillion debt. And now we are seeing almost all the country is sinking for their GDP. And the worst performer is Italy. And like their GDP will shrink 12%. And even including United States, 6%, close to 6%. India will be like a 5%. And Japan, Hong Kong, name any country, except few countries which GDP is not changing, and actually they are getting much better. China and Indonesia with this crisis. So again, this is a case presented by Bloomberg on the end of the May. So this thing keep changing. It can be altered more drastically. And so with COVID-19, what exactly happens? So as you know, human has an immune system, and same way all the living organism has some kind of immune system. Even nematodes also they have immune system. Plant also have immune system. We primarily relies on T cell, B cell, microphage, and fibroblast. They are the core pillar for immune system. So T cell is two type, like a CD4 plus and CD8 plus. So it's a kind of a marker on T cells. I'll try to talk in a layman way so that some of the audience which is not in science they can try to understand. But I don't know how much I can make them understand those who are not in science. As there is a saying that every scientist has a very good story, but they don't know how to tell the story to layman. But I'll try. So CD4 is primarily involved for the B cell maturation, differentiation, and reprogramming for its antibody production. Antibodies, this is what you need for when you are in crisis. So pretty much we need CD4 T cells. CD8 cell is a cytotoxic. It's find out the bad numbers, and they says okay, this need to be eliminated from the cells, from the tissue. So they take care of the bad infected cells, debris, and it's actually potentiate so many signals, and it's also help mature cells, and also there are there are many other function of CD8 cells. One of the characteristic things happen in COVID-19 is that they deplete CD4 and CD8 cells. As you can see, healthy and ICU patients, they just ran out of the CD4 plus CD8 T cells. What happened when you run out of the T cells? It's a crisis. You just can't replace it. Because if you don't have a T cells, how will you go for the immunomodulatory function? Because T cells determines a lot and lot of function for your immune response. And with this crisis, I would like to, as I, one of the primary talk for my, one of the primary goal for this talk is to introduce how this cytokine storm happens. So I'll start with one of the basic slide. It's just recently published by Tate et al. And it says when you have a coronavirus infection, as everyone know, I'm not talking about the anatomy of coronavirus, but coronavirus is a virus which has a crown made of spike protein, S1, S2 and S3. They call S prime. So S2 is S prime. So they have spike-like protein and some multimer structure, and this protein has an affinity with ACE2 receptor on the human cell surface. So ACE2 is pretty much required for normal functioning. It's a angiotensin system, and that's what also involves in a blood pressure related disease and cardiovascular disease, and this virtually present in every cells, and it binds to S2. One of the characteristic of COVID-19, which makes COVID-19 unique, is that it has a receptor binding domain, and it's a very much lock and fit model. It's not the non-specific interaction which virus utilizes to enter the cell and sneak in, and no one showed, like no one knows, hey, from where the virus come. But then it's such a strong interaction that it's just literally enters the cell through the H2. And when it utilizes H2, it also utilizes TMPRSS2 protein. And there is another protein also involved in this whole process, is a purine. TMPRSS2 protein is actually phosphatases and proteases. They help in some maturity of the glycoproteins of virus and allow them to enter. When the virus enter, it replicate. It's RNA strand. It's a plus RNA strand, sensed RNA strand, and it divide immediately. There is no wait time like a HIV. We go there and sit for 10 years as a latent state. It just go and start rolling. It's a amplification start, and so whenever the cell see this virus enter to the cell, like they have their own response, and one of the thing like it's involves a programmed cell death, and the programmed cell death over here is slightly different because it's alveolar epithelial cells, and they pretty much encounter all the time these viruses. So they go a programmed cell death phenomena known as pyroptosis, and pyroptosis is a pretty much involved the inflammasome like signaling. It's a kind of signaling which I'll introduce later part, and it's whenever it's involved, it's inflammasome signaling is pretty high in macrophage. Macrophage handles very well, but other cells don't, because they just do whatever. Like they it goes to one of the massive crisis state, and it's basically call for the rescue pyroptosis, and it releases pyroptosis whenever it happens. Inflammasome related signaling, it releases IL-1 beta, which is a potent inflammatory cytokine, and virus replicate and goes back to the lumen again, which is you. This is what when human exhale, you contaminate other humans. So when it happens below this cell, we have a macrophage. It's always sensing. It's vigilant, and it's saying, "Hey, there's a crisis. So we need to put back some of the call for rescue measure, like when something happened with the crisis, like cop calls to another cop, hey, showed up. Like, um, so there is a tremendous amount of cell sneak into the area where this things is happening, and so they pretty much call T cell, monocyte, macrophage of other origin, because the alveolar macrophage is slightly different, and so there is a two thing can happen when the virus enters, like in the lumen, in the alveolar lumen. So, so the alveolar macrophage, which is just sitting below the epithelial cell, it can start clearing the virus, because macrophage has a power to just eat anything. It can eat plastic, it can eat glass, whatever it can eat, and so one of the unique things you need to know in normal human, like when the things is okay, CD8 cell comes, do a lock and key model like style, like TCR, T cell receptor, TCR, and it's fit exactly to the TCR counterpart, which is expressed on the dying cell, programmed cell death, and Also, it helps and assists by releasing so many cytokines, which is cytotoxic in nature. And this clearance leads to very nicely elimination of your own cell, which is loaded with a virus. And nothing happened, nothing bad happened. Then cell will clear up the space and virus is eaten away, and microphage is also gone out. And if we have antibodies against those virus, it's actually espinos those antibodies coated with a virus and clear off. That's fine. And these people are asymptomatic. Like, you know, they will not have a cytokine storm, and they will not have a symptom. They are doing fine. They even sometimes they don't even have a fever, and they can very well take care because they have a very good CD8 cells. But what happens if this CD8 cell is not able to do their own job because it's got depleted, it's ran out of the receptor, it's ran out of the memory, like it has a memory for those virus. In case of coronavirus, we don't have a memory, so that does not apply most of the time. So it start clearing up all the cells, okay, and most of cells start dying with a later advancement of the cells. And moment the cell is dying, it create a gradient. All kind of inflammatory cells just sneak in and lumen, like macrophage, T cell, monocyte, and they're just fighting with each other. They're just saying, hey, do this, do that, do this. It's like a chaos. Like they're saying that if you make a house, the suggestion of 100 people, you don't make a house, you make a Jew. So it's like 100 inflammatory cells showed up with a different marker saying, do this job, do that job, and just a chaos. And that leads to so much of inflammatory cytokine accumulated in the one place that there is a massive, massive damage. And when it happens, the virus got a chance. It enters sneaking in your bloodstream, and from there, you just don't have much options. Uh, so, so people find the potential therapeutic approach, like either you block the surface protein of a virus so that it can't bind because ACE2 is not available, because hey, surface spike 1 protein is blocked by antibody, which is like a vaccine, humanized vaccines, and there is no virus can gonna sit on surface because ACE2 is not available to access those spike protein. Or otherwise there is a neighboring helper for ACE2, which is TMPRSS2. You can inhibit that through the so many small molecule inhibitor, and then you can cure and just don't let this virus enter your cells. Or maybe just reprogram CD8 cells. You sensitized before through the vaccination program and all those, and they are pretty good with this. Most time it's happens through the natural vaccine-based immunity, and also nowadays is a CAR T technology pretty much in use. Or ultimately you can just clear up all the pro-inflammatory cytokines from your blood through the filtration, and then you call it good. But before I move anything, so I just want to introduce to the trinity of all inflammation. So the trinity, say for example, if you believe trinity as a religion, uh, IL-1 beta, TNF alpha, and IL-6 is the god of inflammation. So IL-1 beta, if anyone has a question, you can post in mid-while, because I understand there will be many uh, non-scientific people, so I can help that way. Um, so you're exempt to just shoot a message, and I'm okay with the interruption. So IL-1 beta is one of the potent inflammatory cytokine, and it's a master of all mistake. So the way it's master of all mistake is that if it showed up, it just destroys things. And the problem is IL-1 beta, you need it because IL-1 beta is needed for the development, differentiation. Well, IL-1 beta knockout mice dies, um, and TNF alpha is also a very similarly potent inflammatory cytokine. IL-1 beta traditionally follows the MAPK signaling, binding with IL-1 beta to the IL-1 receptor 1, receptor 2, triggers signaling through receptor tyrosine kinase. It's IRAC4 phosphorylation, 345 phosphorylation site leads to IRAC1 phosphorylation as 209 and 387 serine threonine, which leads to FRAF6 activation through the monoubiquitination. It's sorry, I'm going to science. And this monoubiquitination leads to activation of TAC, which also is an E3 ligase. So TAC get activated. And then when the TAC get activated by monoubiquitination at 63 site, it's autophosphorylate 184 and 187 residue, which which massively boom for the signal. It's a surge of signal, and there is a one protein known as I kappa B alpha, which holds up all the bad amber, and TAC1 makes sure that it degrades. So this NF kappa, when P65 enters a cell, the first thing it do is IL-6. No, NF kappa B actually first goes and it's overexpress I kappa B alpha, but then one of the things first it targets is IL-6 and IL-8 production from any cells. Similarly, TNF alpha, it's also follow the MAPK signaling, but then it uses a TRAF2 and ASK1, which is MAP3K5. And it goes in a very similar fashion. And it's also the same activation of MAPK signaling through P65 and F kappa B family and do the same, pretty much same effect. But the, sorry, this should be here. So, okay, no, actually, so this should be here. So one of the thing is like, who makes it IL-1 alpha or beta? IL-1 alpha and beta is pretty much same in function, but they are different in amino acid. They are pretty similar at 40% amino acid level, but they are function same, and they both bind to the same receptor. So the biggest producer of IL-1 is actually macrophage. Macrophage makes IL-1 beta from the proform by activated caspase over here. The caspase only get activated when the NLRP protein get multimerized in response to ATP, monosodium urate, virus, any other stimulatory factor, nigericine, and or some other inflammatory signal, which is like mediated through the other cells, like MCSF, macrophage colony stimulating factor. That's a chemoattractant to call for macrophage to rescue, or interferon gamma or alpha. And so when macrophage makes it, it's actually makes for intention. It's IL-1 beta is a potent producer of chemokines and cytokines. So ultimately, cytokine storm begins from the IL-1 beta. When it begins, it's, as I mentioned, IL-1 beta makes sure it start IL-6 production first. On TNF alpha do the same thing. So when IL-6 showed up, it's IL-6 does not do a traditional signaling. Sometimes IL-6 is you needed because it's a bad, it's a good. Sometimes you really need, even people who do exercise, they have a huge amount of IL-6 in their blood, but they are not bad. They are actually healthy and they need for their reprogramming event. And IL-6 is a part of LIF family. So, and LIF family is pretty much involved in the stem cell differentiation. So the lot of cells get differentiated by response of IL-6. So, but IL-6 trans signaling is one of the bad things. So what happened? It's a monomer, and it binds to the receptor GP130. There is a one receptor GP130, and there is another receptor IL-6R alpha. So IL-6R alpha and IL-6 and all, they are tremendously produced by bone marrow cells all the time, and they needed the differentiation inside the bone marrow. But then when they showed up in circulation, it it binds to the GP130 as a homodimer, and homodimer versus like it's a tetramer plus tetramer subunit, and it's caused the trans signaling. Like you can see over here, IL-6 bonds to the GP IL-6 receptor, and then it binds to the IL-GP130, and the signaling goes on. When this state reach, it follows the STAT pathway, JAK1, JAK2 autophosphorylation, followed by STAT3 phosphorylation at 7057 subunit, and STAT activate the STAT dependent gene expression. And so nowadays, in rheumatoid arthritis, there's so many inhibitors which is successful against JAK2 inhibition. It's known as tofacitinib, is one of the best known drug for rheumatoid arthritis. And another one is antibody against IL-6 receptor. Moment you take out the IL-6 receptor, IL-6 won't bind to the GP130 because it's need heterodimerization. So it's known as tocilizumab. Tocilizumab is one of the biggest money-making drug for Genentech, Roche. So it's like billions and billions of dollar economic done by this drug, and it's used worldwide. And it's one of the best known biological available for rheumatoid arthritis treatment. And I think 70% time successful to the in the disease etiology. So before I move, I would like to let you know why COVID, which is the thing which I'm talking about, IL-6, why COVID does not affect much to the children. It's my own interpretation. So it could be changed with a scientific knowledge, which is happening every day at the record pace. So the thing is, when the children, this is the gene expression profile for adult versus infant CD for effector cells. I didn't find much data available for the lung tissue. So, but this data was very good and convincing. This is published in JEM 2017. Yeah. So basically, it says if you see infant has very low amount of IL-ST, it's a GP130. It has a very low amount of IL-1 receptor too. So there is a very low chance it will have a IL-1 signaling. If IL-1 signaling is low, it will have a very low chance that IL-6 will be produced. Even if it's produced, it has a very low chance that the signaling will be triggered because there is a low amount of receptor for IL-6. So pretty much, it's a it's a it's a advantage. The whole situation put them in advantage that they, even though they exposed to virus, they always go to the asymptomatic way, and there will be no damage much to them because they don't have much things to be played by cytokine storm. It's my interpretation, so it can change. So what happened when it like IL-6 based analysis done by some 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 of the people who done this IL-6 based analysis. One of the finding I would like to tell about. Uh, I was reading about this COVID-19 thing come from the China, and they did one study, the Wang et al. It's published in BioRxV. Sorry, I forgot to mention this paper, but I should have put it. And just it's slip out of my mind. That paper has inspired me a lot. So one thing they showed in the Wuhan Cancer Center, it's not a typical Wuhan medical something. There was like a 69 patient admitted. So 69 patient admitted, they found the two things happening, and it's happening in the January 15 to January end. Is a span of 2 weeks this study happened, and this is like the two kind of a patient admitted, one which has a very low oxygen level in their blood, and one which has a little more than 90% oxygen in their blood, and they call it good. So they treated those patients with the antiviral medicine, and they got recovered. But the worst performer was those who has less than 90%. And there were 14 patients among them who has very low oxygen level. Sometimes oxygen level low level can reach to 50%. And I don't know how people survive at the 50% oxygen level because the most of organs supposed to die at that level. And then they found out of 14 people, 5 died immediately. Now, it's like no way. And though the one of thing characteristic thing about they observed that they have like a highest amount of IL-6 in their blood. And IL-6 supposed to happen between 1 nanogram per ml to 5 nanogram per ml. That's a normal range. 5 nanogram is also very high um, for the systemic inflammation, but it's supposed not to go more than that. They found even 16 nanogram per ml, and there are a few cases in COVID-19 patients like it goes to the 200 picogram per ml, and this is a very alarming high IL-6. So they also screen many things, and this has also TNF also alpha they found correlative. But at the same time, the study in parallel happening throughout in China, in a Wuhan area, Wuhan district, and they found only one thing common among all the. Somebody says IL-10 is also there. Somebody says TNF alpha is also there. Only common thing was IL-6, 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 IL-6. That was a consistent between the all result in the all the human cohorts, and based on the IL-6, now people have defined IL-6 and COVID with a low IL-6, medium IL-6, and high IL-6. High IL-6 is pretty much like 90 nanogram per ml, and you you'll be surprised that moment COVID, even the COVID patients, based on the IL-6, their proteomes are very different. As you can see, more and more coagulation related factors are enriched with the highest amount of IL-6, and their immunoglobulin and immunity is also changed. They have a very typical immunoglobulin proteome showed up like a light chain. They have a very much variable thing going on in light chain in immunoglobulin, and which always affects immunity. And they have something like you can always see those PCV analysis. They how they are look very different based on the IL-6, and they are all same COVID patients, but then proteomes clearly dictates that IL-6 dictates the terms for COVID-19. And since I'm a rheumatology researcher, so always fascinated by rheumatology finding. So Mokuda, this guy is from Japan, I think. They found that actually, if IL-6 is present in the synovial fibroblast, exposed to synovial fibroblast more and more, it produces more and more ACE2, more and more ACE2, more and more COVID binding, more and more COVID binding, more you exposed to virus, and that thing can be also abrogated, inhibited by STAT3 inhibition, which is IL-6 signaling, the part of IL-6 signaling. And one of the interesting data from the New York, when this data, when I'm talking, is like there was like 22,000 confirmed cases. This is the last month. Is based on again uh, age-related issue, like old age people get more and more COVID, and it's pretty much true. But then one of the thing was uh, among the different race, surprisingly, it seems like one of the two-third people got discharged. Those who go to ventilator, you know, you know, there are many people who goes to ventilator. The moment you reach to the ventilator stage, possibly only one-third chance that you die, or two-third chance you'll survive. They found one of the alarming that Caucasian race is 50-50, means out 1 hour, 1 out of 2 might die. So it looked like this COVID infection is also based on the race, affects differently. And so as I mentioned you previously, so you really want to make sure that your CD8 positive cells are pretty much healthy. And COVID-19 infection wipe off CD8 cells. So what happened if the IL-6 ratio is correlated with CD8 cells? You'll be surprised if you can still survive with so much of IL-6, low IL-6, but if you go if you lose the CD8 cells, here is the mortality rate. It's a Kaplan-Meier curve, and that look very detrimental. So IL-6 in combination with CD8 could be the new marker to predict the morbidity in hospital setting. And based on the infection, like there are two people. One is sorry, I'm still taking a little longer time on my slides. So there is a asymptomatic people and symptomatic people. So asymptomatic people is pretty much mm, don't have any sign, and they have not they have a longer, very less immune response against COVID-19. Their blood titer won't be positive for COVID-19 antibodies, and they, as I mentioned, they don't get cytokine storm, so they don't have a very efficient uh, immunological reaction going on. So even their plasma will not be used for as a convalescent plasma as a donor, and but then they carry coronavirus for longer and longer time. And one of the characteristic of COVID is they have a ground glass-like opacity in the lower lobe of lung. And ground glass is pretty much a cellular infiltration. And this cellular infiltration is, we have a mice model for infla, infl, inflammasome or inflammation. It's known as a air pouch model, where we just create a pouch and then pouch with a monosodium urate, and then there's a tremendous amount of cellular infiltration happen. And its annual model is animal model dictates how you can actually cure the local inflammation in the pleural area. And so this ground glass opacity determines the severity of symptomatic versus asymptomatic. And as I mentioned, symptomatic people has more IL-6, as you can see from this slide. And then I went ahead and analyzed some of the data, which is common. Nowadays, people think there is an 18 cytokine, which is the characteristic for COVID-19. And this amount, I put in some of pathway analysis, and I realized when I put this 18 unique characteristic, it's hints that it's all involved in a cytokine receptor interactions, leukocyte infiltration, enhance survival through the AKT signaling, um, and many like monocyte regulation, cell migration, and they all so closely tied up. All the networks are so closely tied up. And one of the unique thing which I found from this analysis was the highest candidate was a rheumatoid arthritis hit. I said, hey, wait a minute. It's not a rheumatoid arthritis. These candidates are characteristic of the COVID-19. And I says, well, the rheumatoid arthritis is an inflammatory disease, and it's full of so much inflammation. That is the local environment is mimic exactly the lung local environment for the purpose of inflammation. That make me puzzled. I said, wait a minute, rheumatoid arthritis, I can think about it, rheumatoid arthritis. And so like other diseases like Crohn's disease and they atopic dermatitis, they are all inflammatory diseases, and very similar to the each other in terms of cellular damage, tissue damage at different places. And one of the things which I found was very characteristic is like what are the similarity, like COVID is infection, RA is autoimmune. Target tissue, alveolar membrane. Rheumatoid arthritis is synovial membrane, both related to membrane. Smoking is a risk factor. Yeah. But then nowadays, COVID-19, smoking is not the risk factor. Well, many publications says. Uh, so disease is acute versus subacute. It's a lethal versus chronic. Glucocorticoid, it's not determined. So basically nowadays it's find that it's actually helping. So then IL-6 production, TNF alpha production, MCSF, macrophage cholinesterase factor, IL-1 production, IL-17 production, they all similar to the rheumatoid arthritis and macrophage defense system, uh, peripheral monocyte cells and T cell response and fibrotic response, they are pretty same because it's again a membrane related disease. So what exactly happened? I have shown you this slide previously also, very similar. But one thing you need to know here is it's the dynamics of cytokine storm. So in this dynamic, what happened? Lung has two kind of a cell: alveolar type 1, alveolar type 2. Alveolar type 2 is ciliated. Alveolar type 1 is flat. It's epithelial morphology. Always below the lumen, there are so many macrophage. He's like a police guard, watchman. And when it happened, I mentioned you, when it happens, program cell death happen. And if they die nicely with a program death without apoptosis, cell, the whole macrophage will take care. But if it's not, then it's worst. So what happened is like when the cell death happened, it's a dendritic cell uh, takes up the information, present things through the uh, MHC2 because antigen presentation, and CD8 and CD4 plus cells showed up. Help this to know this is which kind of antigen. Ask B cell to make something. B cell makes antibody. When B cell makes antibody and very specific, you clear the virus. That's fine. But the problem is if the damage goes on, like this particular damage goes on, there is a there is a no gatekeeper. The gate is open, and so there is all kind of a cell enters. Like even they call microphage from the bone marrow and says, "Hey, bone marrow, just save us." So all kind of a cells enters. They like make the battlefield, creates a tremendous amount of local surge of TNF alpha, IL-1 beta, GM-CSF, IL-17, IL-10, name them. It's like 18, as I mentioned you. But then you didn't see one of the most damage happen in this call of rescue is the loss of this layer. And this layer is open up. When it happens, the fibroblasts sneak in because only fast way to recover is the fibroblast. Fibroblast makes fibrin, and they just patch it's like a patch, and they patch it up so that nothing should enter, and uh, and that's the worst problem because when the fibroblast comes in, it's create a fibrosis, and fibrosis is a loss of capacity of lung, and you are filled with a liquid, you are filled with a fibrosis, fibrotic plaques, and just you can't breathe it. Even the ventilator is not helping because there is no gas exchange. There is no epithelial cell which can exchange the gas, and people die because of no oxygen to go through the lung. And in rheumatoid arthritis, also very much similar happens when there is a scarcity. This fibroblast makes the fibroblast like cell, which makes the so much thickening of the tissue. It's known as pannus. That this pannus cause more inflammation and rigidity of the tissue and structure. So when I mentioned arthritis, rheumatoid arthritis, all the cell player, can we really utilize the drug against the rheumatoid arthritis? Yeah, there are plenty. Like we have a drug against this APC and T cell presentation, ebacept, and T cell to B cell interaction, rituximab, and TNF alpha inhibitor, which is like one of the Enbrel is one of the first kind of a medicine, and they're all owned by different companies. And IL-6 receptor inhibitor, which is I mentioned the tocilizumab, one of the biggest minting for money making for pharmaceutical industry, and then there is an inhibitor for IL-1 beta, like canakinumab, and rilunacept, where the Purushottam works. That comes from Regeneron, rilunacept, and JAK1 inhibitor, tofacitinib, as I mentioned. So all they seems to play all kind of a part of the role. And can we really think about it? So one of the new, just 3 days back, I read that coronavirus breakthrough dexamethasone is the first drug shown to save lives. That's actually one of the anti-rheumatic medicine. So look like rheumatoid arthritis medicine can help. Um, so now there is so many clinical trials going on. Like you can see the tocilizumab and um, IRAK inhibitors, canakinumab, like an NLRP3 inhibitor, as I mentioned. Over here, there should be a rilonacept also. Sorry, I didn't mention it. So TNF alpha inhibitor and MCSF receptor signaling inhibitor. One of the company I know, Kineska, they have they have very good success with MCSF inhibition and patient recovery, and their data is yet to be out. And IFN gamma inhibition and JAK-STAT inhibition, just like as I mentioned, tofacitinib is one of the best known medicine for the STAT inhibition. And so when I talk about one of the best known medicine for rheumatoid arthritis, tocilizumab, although I'm not trying to glorify that medicine, I'm not the uh, I'm not the advocate for that medicine, but then yeah, it's a good medicine. And one of the things which you see is like it stopped IL-6 receptor to bind to the surface, so there is no IL-6 signaling. So there was one paper showed up, I think last week, and it showed up that if the patient gone with a tocilizumab treatment, look how they recover nicely. Like they have a gene expression profile, and there is so much of inflammation going on, like TNF, CCR3, IL-1B, IL-6, IL-10, and when they compared with the healthy control and the recovery stage, how nicely they are recovering. And apparently, it is getting successful. But then there is still a dispute going on whether it can be part of it can be used as a drug, because it's still need of further very nice clinical trials throughout the world. And unfortunately, India has not done any clinical trial on tocilizumab. China is doing, US doing, Europe is doing. So and some of the vaccine which you expect, one of the people might know Moderna, like mRNA based therapy 1273, and people are thinking of adenoviral mediated. Some are just want to put the DNA plasmid against spike 1 protein. Some want to modify the dendritic cell through the lentiviral infection and correct in the human patients. And some are just want to modify this antigen presentation cell so that we can recognize more. So this vaccine development is a ambitious program, and it takes time. I'm sure by next year it should be there. But one of the challenge is this virus is mutating faster than actually HIV timeline. So it's it's still hope, but let's see how it goes. And there are the world statistics how there's so many phase things going on, like exploratory versus now the phase one. So now this is the prime thing you should focus on, because this is already validated with this much, and this phase one is a good hope and expectation for the cure. One of the interesting thing which I would like to talk about this COVID-19. I'll try to finish in 5 minutes because I'm gonna reach 10. So one of the things which happens in COVID-19 is why male dies than a female more, and literally 1.7 male dies compared to one female. So it's like a two-third male. Is it a male specific problem or is a female specific problem? Um, so there is a gender bias literally look like. So what makes this gender bias? As a layman, I'll try to explain you that as everyone know, female has X and X, 23rd chromosome. Like there is a two copy of X, and male has only one copy, including Y. So Y is matched with some part, but then there is some part doesn't match. So since woman has two X, nature don't want this two X to work all the time. So they do an inactivation of one X and says, hey, one X, you need to shut the mouth. And one X need to work because male has a one counterpart X, which is similar. So it's known as meiotic sex inactivation. So meiotic sex inactivation determines a lot of things in the woman. Like it's also dictate the term for their uniqueness, their metabolism, and since dosage for X and X is compensated between men and women, so they are pretty go head to head. But then one of the thing uh, which makes them different is since everything is programmed through the estrogen, and estrogen is an immunomodulator. Male counterpart is androgen, and androgens are not that immunomodulator. It does, for the context of the cellular proliferation and all those things. So one of the things is like ACE2 is pretty less in women. Like if you see, is down-regulated by estrogen because they go on an estrogen cycle, and ACE2. So they are having less and less exposure to virus because there is less virus to enter the cell, and they also found, like people nowadays found out that another counterpart TMPRSS2 is androgen regulated, which is also a disadvantage for the men. But one of the unique thing which they found was TLR7. As I mentioned you that virus utilizes any danger signal utilizes TLR signaling, and it's a TLR7 signaling. So one of the thing is like as you can see, Mitotic X inactivation is a lose sometime, and not all the genes are inactivated all the time in another counterpart of X chromosome. So what happened is like sometime some are leaky, and one of the leaky gene was the TLR7. So the TLR7 is pretty much not stopped by dosage, and they go like greater and greater expression in the female uh, innate immunity cells, and they can recognize the virus much better and much faster and much quicker, and they can grab this virus, they can process immediately, and the men lose this time, and so that has an advantage. And also people reports like why old people catch the COVID is like because of the lack of B cell activity, because they can't produce that much of antibodies. And one more thing is like in COVID-19 there is no long-term memory, people speculate, because this virus does not go to the germinal center or thymus or spleen where is to antigen presentation, where the memory cell is developed. So there is a very less likely that we will have a prolonged immunity for COVID-19. And so it's postulated, data is still speculated that it may have some good amount, a year of, you know, year of immunity, maybe. So that's put women in advantage compared to the men for this disease. And as I mentioned, the children has advantage. That's again my interpretation, that because they have a very less likely inflammatory signal to develop. And with this, I'm thanking CoronaCure for asking me to organize this whole thing, and I can take any questions.
Okay, thank you, Dr. Ani. I think that was really insightful. A lot many things we were not aware of, and I feel it has been a lot insightful to us and more of biology. I found it interesting. So we'll go with the questions. We have one question here right now. So it's by Mona Lisa. She asks, can we use MAP2K4 inhibitors to down-regulate IL-6
Yeah,
signaling?
we can do that. But then problem is, it's not only IL-6 determined by the MAPK. IL-6 is the inflammatory response done by the macrophage, and it can be triggered by so many TLR pathway. And IL-6 sometime you need, because problem with IL-6 is that B cell needs it. B cell feeds on IL-6, and B cell required for their antibody production and immunomodulatory function for on IL-6. You can't, you don't want IL-6 to completely shut off. That's why tocilizumab is a dicey thing also. Although it's happening, but you don't want to completely shut off IL-6 signaling, but IL-6 production. But yeah, you can modulate them. And it's actually IL-6 makes IL-6. So this best strategy is to shut STAT signaling, not the MAPK signaling, because MAPK is a survival and it's immunomodulatory. Possible strategy is the STAT, I would say. But yeah, you can still get the same effect, because I worked on natural compound like EGCG, and we have shown like MAPK signaling shutting by EGCG, and the TAC, and even in the gout helps the disease disease etiology. Yeah, did I answer?
Yeah, I think so. Then there is one question. If I'm reading it, this is from Dr. Sonali. She is asking, are there any evidence of virus using HSPG to dock before ACE2 interaction?
What is HSPG?
Uh,
Hello, can I speak?
yeah,
Can I
sure,
ask my
please
question?
go. Yeah, please.
Yeah. Okay. Thank you so much, Dr. Anil. It was great presentation actually. And so my question is, is there any evidence of the SARS-CoV-2 is actually, using HSPG, which are heparin glycan sulfate, before,
Okay.
before
Yeah.
binding to the ACE2?
So the spike protein is a glycoprotein. Okay. So the
Correct.
glycoprotein,
Yeah.
targeting glycoprotein is also a big strategy. Like in our lab, we always work on some of the like some of the protein like syndicant. And one of my colleagues works on sulf to ruby, and she also, we always target this glycoprotein related thing, but it's a lot of challenge. And one of the thing is like sometimes glycoprotein is needed for the activity of certain thing. Like even tocilizumab is glycoproteinated for the better function, and so you have to determine very good way, whether you want to make the bitter as glycoproteinated, strong, bioactive. It's a big challenge. And does work. Uh, like I know ACE2 is glycoproteinated. If you make this ACE2 less glycolated, you lose the interaction with COVID-19. And there is a target against this. There is a mm, two medicines in clinical trial against ACE2 glycosylation. I don't remember. I could have put that one. So yeah, did I answer? I don't know.
Yeah.
Okay.
So we'll move on to next question. This is from Arif. No, you have? So I'm so sorry.
Yes, sorry. I actually, my question was little different. I was talking about the HSPGs, which are not the ACE2 ones. These are also present on the, the lung cells. These are like, kind of heparin glycan sulfate separate from the ACE2 glycosylation, which are generally required in case of binding with heparin. And also there was a study that they were actually, the last SARS-CoV in 2003 uses HSPG to dock first, orient itself, and then goes to ACE2, binds it, and then actually gets internalized. So do you,
Go ahead.
So my question is that, do you think, I mean, it was just one study, and that too is talking about SARS-CoV 2003 one. Though I did a lot of course, there are a lot of studies also come up that SARS-CoV and SARS-CoV-2 has almost 90% similarity on their S protein side mostly. So I'm assuming that, this would also this would also follow the similar pathway. But are there any studies, or I mean, what's your comment on that? Do you think it's also possible with SARS-CoV-2?
Yeah, that's definitely possible, because the HSPG are the micro modifier. So moment HSPG dynamics change, so it changes the landscape. And so HSPG primarily responsible for chemo attraction. It's not the cytokine mediated signaling. It's not the inflammation signaling.
Yeah, absolutely.
So the problem with the HSPG is like before, when the cytokine storm hits, cytokine overtakes it. The chemokine-like function, because anyway HSPG is involved. And also there is so much of ambiguity about this microenvironment related thing in SARS COVID, because SARS and COVID is like the problem is there's not well studied to the microenvironment. It's too dangerous to work. And
yeah, I mean, actually, I was just thinking, I mean, it's actually pretty much even before the entry of the virus, it's actually at the membrane. So if one inhibitor can be, used for HSPG or to block, to do an binding inhibition, competitive inhibition with the virus, where the molecule can go and bind with HSPG so that it doesn't get any docking site, and it's kind of prevented the entry of the virus itself. So I don't think then we do see the cytokine storm only happens when the virus already enters into the cell, right?
Yeah, but then one of the things you have to remember, this virus is unique. This no one have seen this virus in nature much before. So other viruses such as HIV and all this, even influenza, they utilizes non-specific interaction. In non-specific interaction, HSP play a lot of role. You know, one of the things
Hmm.
is Cytokine 2 and 4. One of my colleague works, and but the problem is it's targeted. There is a receptor binding domain for virus, and it's such a unique and so strong binding that it just catch you. You know, it's like ACE2 is there, it catches ACE2. So none of the virus is so unique in binding their potency. Like it's one of the highest potency binding virus. And even though if we try to modulate things through the HSPG determines the charge, you know, charge of the on the extracellular surface, and and moment the charge is more and less, it's calls for the infiltration differently. You know, they call for those cells like monocyte and other cells very differently. But this virus can sneak in anyway because it has a receptor binding domain which is beyond the understanding of our chemokine and HSPC knowledge. It's a modulator, but it's not the sole determinant.
So you think
HSPC.
even we could prevent the binding with HSPG? There are still chances that virus can go
Of
pretty
course.
much.
Yeah, pretty much because now it has a it has a one protein it utilizes is ACE2. Even though there is a no TMPRS2, it will enter the cell, and that's why the clinical efficacy for this hydroxychloroquines and all working fine because it's changes the pH after binding. So one of the biggest strategy for curing this virus is actually why we are getting failed is that we are the whole world is just behind spike protein. They are just developing vaccine after vaccine. There is a everyone is making vaccine against the spike protein, but you can't make the vaccine against the spike protein because it's getting mutated and it's so specific that it's very hard to determine the receptor binding domain. Receptor binding domain is not readily available outside to actually entertain antibodies. So just catch and people are not focusing on other parameters. Like as you said, HSPG could be modulated, but I doubt it can change a much in terms of infectivity. It may change 70% or 30%, but still gonna be infective. And
So
changing
so you
HSPG
mean Mhm. So you mean it could reduce the viral entry, but it would still happen
yeah,
if there is a HSPG inhibitor is also present. Yeah,
yeah, because it's a very specific, it's a ligand based interaction. You
correct.
have to this virus is never been seen in nature. It's very unique. It's very unique. Humans have never
Hmm.
seen it.
And also
Okay.
the other,
Uh,
sorry,
Sonali,
am I?
Sonali,
Yeah,
I'll just ask few more questions over there, then I'll get back to you because we are
sure,
having
sure.
little bit of time.
Please, yeah, yeah. Okay,
Thank you,
thank you.
Sonali.
No problem.
Sorry for that. Okay,
No
so
problem. Absolutely fine.
yeah,
Thank you.
so yeah, Dr. Anil, we have few more questions from Arif. We have a question like in vitro simulation of PBMCs with peptide pools, they didn't show much of significant difference in IL-6 level in different groups. Is really you think that IL-6 is a cause behind cytokine storm? It's a recent
No,
work.
no, no, no. As I mentioned, you mentioned before, IL-6 is a part of the trinity. It's just my term. Like you know, IL-1 beta is the master of all mistake. It's start things. IL-6 can take over. So IL-6 found to be more correlative among all the patients throughout the world. It showed up as one of the greatest marker. There is an 18 player in this whole game. Like it's a cricket pitch. There is a 18, 11, you know, there are a lot of player in this. IL-6, even if you suppress IL-6 signaling, yeah, it does improve something, which is JAK-STAT inhibition. Uh, but then I don't think so. Only IL-6 is the target, but yeah, IL-6 does play a lot of role because, as I mentioned, it seems working for children and seems working for these inhibitors, and it's taking care. And there is no straight there is no straight answer for this actually, because there is no one gun which just kill all the birds. So there
Yeah.
is a chance that it might not show up anything. And IL-6 can be always necessary for the body also. So
Okay, so
that's
look,
possible.
so actually related to that only, the other person, Tarun, he asked like, there are other infections where you, as you had said earlier as well, highlighted that IL-6 is already circulated, those
Mhm.
inflammatory diseases. So he's asking whether is there any possible mechanism in the body to divert or redistribute such IL-6 responses at different sites?
Uh, so IL-6 play a very good role in terms of um, as the signaling code required. Some places IL-6, as I mentioned you, IL-6 plays very good when it's in a close contact with environment. Like in bone marrow, it's needed for development. T cell maturation. Now B cell also needs it for their antibody production. It's a modulator. Now some of the distinctive signaling come from the trans signaling, where it require IL-6 receptor and GP130. So it's subjected to which kind of a signaling there's a surge of IL-6 signaling, and it's happening throughout the body all the time. But characteristic is you should not have highest amount of IL-6 and IL-6 receptor in your bloodstream. That can change a lot of things. And also, whenever you have an infection, any infection, any microbial infection, you'll have a surge of IL-6 in your blood because that determines the fever. So again, it's a immunomodulatory cytokine. So and it's a did I answer or just I went wrong?
Probably. Yes, because I think what he meant is like how you redistribute IL-6. So basically you answer saying that IL-6 receptors can be regulated. So that is one thing you have answered it. So another question that we have is, what could be the possible reason, as ACE2 is a common receptor for many coronaviruses like NLP6 as well, or other coronaviruses. So
Mhm.
he's like, what could be the possible reason for massive cytokine storm, especially in case of COVID-19, not other coronavirus family infected patients?
So it's a lytic cycle. Whenever it hits the cell, it divide very fast, and so cell can't keep up the pyroptosis phenomena. Pyroptosis is a call for help, but then and it's mediated through NLRP, and it's so blind that more and more cell dies. It just throw up the ATP outside. More and more ATP, it just ask macrophage to produce more and more IL-1 beta, and it just it's just the nature of the virus, and it's so strong interaction that you will not literally see coronavirus flowing in the blood. That's why most of the time the titer showed up, titer not showed up, RT-PCR fail, because it's not floating in the blood. It's just infecting cell after cell, cell after cell. So it has a it's just a, if you say in drug KM, is PKPD. The PKPD of the virus is literally very strong. It just keep infecting cells, and this nature is a detrimental. And the place where it's infecting, it's actually start from the alveolar epithelial cell, and it's just the nature of the virus, and the problem of the strong interaction with ACE2. And one of the thing it's found that there is interesting thing is happening is like, although people say that coronavirus ACE2 is a connection, but sometimes what happened is like, moment the coronavirus hit in the cell, even the ACE2 got down regulated, and then that should be advantage, right? But then angiotensin converting enzyme get activated, because the moment you don't say see ACE2 is think that I need to convert this angiotensin, and then it become a problem again, because more and more ACE2 is activated and available for reinfectivity of this virus. So it's like daemon is amplifying over and over and over. So it's tricky for this virus.
Okay. Okay, so I think there are no more questions in the chat box. I have one question. I found that graph interesting. I'm not being currently being a racist, but I found it interesting, as you said, that the white people or the Caucasian ones have a 50-50 chances of being having discharge or being affected badly with the disease. Why do you say? Is it related to the melanin production, and how is related to the immunity of those people?
No, there is a see melanin is something to go with the skin, but then I don't think so. That's a reason. So again, is there in gene and genetics? So we human being are all similar, 99.99%. The only 0.1% makes a difference, and so that that answer I really don't know what makes them unique. Is there genetic makeup? Is it because of genetic makeup and the way you eat the food? Because the food habits are different, climate is different. Like one of the classic example I can give you, autoimmunity, um, like MS and rheumatoid arthritis, is primarily happen in a very cold climate place. Like many Russians are having rheumatoid arthritis. So it's just because the place where they live and their genetic makeup. So, so that
Okay.
that's subjected to a further debate. Like we need to find out why.
Okay, so we have one question from Pip Banerjee.
Yeah.
So Pip is asking, since the T cells reduce in COVID patients, as you showed in initial studies, are these the peripheral T cells or the tissue resident immature T cells? And related to that, do thymus atrophy during infection has been looked at?
Oh, that's a good question. That's very specific question. So, okay. So the thing is, lung. Lung is a battlefield, right? And the thymus is a reservoir, right? And and uh, uh, and the bone marrow is the National Guard, right? Like National Guard out in India counterpart is CRPF. So 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 something happened. Police goes, and so that's lung T cell. They try to handle it. Okay, it's gone from their hand. It they says we need to send the memory T cells. We need to find out this virus. We have an imprinting of this virus. But then hey, we have the humanity have never seen this virus. We do not have imprinting of this virus on our gene. Our fathers, grandfathers, no one has seen it. So memory T cell, even the imprinted common combination, cannot be showed up. Cannot be showed up. Then thymus try to send some very close by, and so it's atrophy. Thymus try to pick up clonal selection. Then there is a somatic hypermutation things going on. Which one to go? Which one to go? It does happen, but it's a misfire. Again, thymus also doesn't know. They have never seen this epitope, never been imprinted. So then there is a call for CRPF or uh, like hey, which is blood marrow, bone marrow. Then there is a constant surge of natural selection, clonal selection, which could it solve this problem? But the problem is we don't have the best fit. So there is so much of immune response not only happening in the lung, it's happening to the thymus, it's happening to the bone. It's a constant demand. But the problem is there is no epitope selection. So it's because of there is no epitope selection, CD8 creates a non-specific response. And non-specific response triggers like CD4 plus in this case. Non-specific response triggers non-specific B cell production. B cell also doesn't know to produce whatever because it's a non-specific and so much of somatic hypermutation happens in the local milieu and then sometime in spleen, then it causes another kind of a damage to the lung. It's known as antibody directed damage, because they don't want to handle this, because more and more antibody showed up, its complement is going to show up. It's another fighter. It's another kind of fighter. So because of so much of innate non-specific response, we damage ourselves. And I don't think we actually can actually present the case. But indeed, what happened is like this: when moment this coronavirus hit the body, it start getting colonized. That time is a time when we get a very good titer, and this plasma, which is convalescent plasma, sorry, uh, is used, and possibly those human being has read the immunological things much better, and possibly can surpass to the germline as the imprinting for our next generation. Did I answer?
Yeah. I'll move on to a last question. It can be asked by Sonali. Sonali, you can mute yourself if you want to ask the question directly.
Yeah, sure. Thank you for allowing me again, ask question. Thanks a lot. Actually, it's quite interesting and engaging session. So yeah, so my next question was actually, if someone wants to prevent the entry of the virus, how the molecule, I mean, if there is a molecule been designed to either block the HSPG or directly bind to the virus so that it doesn't go and bind to the ACE2 receptor, what should be the route of the molecule in the body? How do you see oral administration or an inhalation or a spray? What is your take on that? Because you said that there's hardly you don't see much virus in the blood. So the viruses are mostly there in the apical surface of the epithelial, right? The lung epithelia.
Yeah.
So sorry, could you?
Yes. Yeah, you're right. Yeah, go ahead.
Yeah, so then what route you think would be the best for getting for any molecule to go and bind to these viruses, if that is the strategy we want to follow?
So if you see there is a damage in the lung, uh, COVID gives us ample of time to face the damage, get damaged, and or cure me without damage. Without damage. So the moment damage happen, the fibrosis will happen. People will die by fibrosis. People will draw by their own, and they die. So and then there is so much of risk factor. Uh, the problem is you have to cure much before. If you really want to stop this thing, that can be cured, and that should be done before. So there could be many wild theory from my side, which I'm not saying that people should do it. So nowadays, if you see this, Lima is a monovalent antibody they are using to generate this COVID spike against spike protein. And moment you inhale those antibody and you see this virus, and the virus get neutralized by antibody. The moment the virus is bind with antibody, microphage can kill it. Microphage got a shot is the best archer, one of the best archer. So the best strategy is we should have an inhaler of those kind of a thing, or maybe nowadays the microRNA is also used for therapy. Like we should have a single stranded RNA as an inhaler. You just inhale in a lung, stay for 24 hours, and roam around. Even moment virus hits, microRNA wipe off. It's a double stranded opposite of the strands. It's a plus strand, negative strand.
Yeah.
It's done. Some of the inhalable monovalent antibody is the best strategy. If you really want to check at the level of inhalation, and you can roam around happily. So that's still doable. But then problem is world is behind vaccine.
Correct.
So
How do you see about a protein kind of molecule? I'm not sure whether a protein can be made as an inhalable
yeah,
kind of stuff.
Yeah, it's inhalable. Yeah, it's a therapy nowadays. MicroRNA, inhalable microRNA is a therapy. as you can see in even vaccine trial, microRNA, Moderna is a microRNA. You just put the synthetic RNA in your tissue, and this RNA start making S1. That's it, and uses the whole thing. But you can also reprogram your body in terms of the success of all the infection lies less and less inflammation, less and less. So one of the biggest finding is like why the smokers are not finding correlative. People say, hey, this virus is gonna kill all the smokers. They said initially, and then they find there is no smoker is dying. You know why? Because the cigarette has so much of anti-inflammatory content, like a nicotine. It shut the inflammation. The moment you are good in shutting the inflammation, either through IL-6, IL-1, whatever the way, you actually cures it. Because inflammation, shutting your inflammation is the key. Like I've seen, like in India, my sisters, they drink. My sister said she always drink haldi, that's curcumin, and
Yeah.
which makes the body good. Yeah, definitely it should work. I work on green tea, so that works. So keep yourself inflammation free, and then possibly you can fight it. And even the inflammation is a success, key of success. Or some of the therapy which I said through the inhalation, like a aerosol based, like a inhaler based, it should work. And peptide based, I can't say because ACE2, you can put against the ACE2 receptor binding domain, but ACE2 constantly get internalized in the cell and recirculated back. So the PK/PD for the peptide, bioavailability of the peptide is always a constant challenge, but it could be a therapy too, definitely.
So,
Okay.
so you're saying that getting a very high concentration at the localized site could be a challenge?
No, bioavailability.
But when you're
MicroRNA,
giving it through inhalation, then it directly goes through trachea and reaches to lung, right?
yeah, and sit there.
Correct.
Mm,
So,
and
so the concentration could be really maintained by giving multiple times of by inhaling multiple times.
yes, that's right. Like I'm not a pharmacodynamic person, but then it should stay for 6 hours. 6 hours
Yeah.
at least.
Okay.
Whenever you go
Okay.
to a high
Thank
traffic
you.
area, just let it go.
Okay, so Dr. Anil, if you are okay, so we can share your email ID with some of the people who are actually very curious, and
Yeah, sure.
I think they can write you email because it's already, I think, 11:30 or 11 o'clock in India, and so and there are questions. There is no end of the questions, and this is a very interesting topic. So for sure, people would keep asking, and there are more curiosity about the idea. So, but I think I would like to call off the session for the day, and I would like to take this opportunity to invite our audiences to visit our website, that is coronacures.co, and the cypreneur.com, and check all possible resources and news and various new information that we are updating timely, and we try to make full use of it. And if you have any other questions, so you can even send us directly to us, and we will for sure try to answer them as soon as possible. And the email ID is teamcoronacures@gmail.com. And also the recording of the today's session would be available maybe in 1 or 2 days on our website as well as on our YouTube channel. And I would like to see you in our next session with Dr. Soman Basak from National Institute of Immunology, that is from India. And on 20th June, around 8 o'clock in India time and 10:30 EST time. So finally, I would like to thank you all for your time, patience, and great curiosity. Thank you, Dr. Anil, for your time, efforts, and such a wonderful talk. This was really great talk and very much interactive talk.
Thank you, Dr. Anil. It was really wonderful, actually. It was really
Yeah.
an interactive session.
Yeah. So thank you, everyone. See you maybe after 7 days. And stay healthy, stay safe. Take care.
Yeah. Stay safe.